Welcome everybody, thanks for joining today's session. My name is Pia Rautenstrauch and I'm a PhD candidate in the Computational Regulatory Genomics Lab of Uwe Ohle at the MDC in Berlin. Today, I want to introduce you to the model LIAM that I developed to tackle the complex integration tasks that are posed by multimodal single-cell data. So let's first take a look at the data that I'm working with. Multimodal single-cell data is obtained through sequencing-based assays that allow a researcher to simultaneously profile multiple molecular layers or so-called modalities per cell. And such type of assays have really matured a lot in the past few years and are becoming increasingly commonplace. A very prominent example for such type of assay is the simultaneous readout of chromatin accessibility and mRNA expression or so-called gene expression in the same cell. And this is also the use case that I'll focus on for the rest of my presentation. A major reason for the sustained interest in such type of data is that it promises to provide a more comprehensive and accurate description of cellular state. And I brought along a biological example for a situation where this joint modeling can actually be beneficial on the right. So this is a schematic depiction of the process of lineage priming, where changes in chromatin accessibility that are depicted here by the color of the nucleus are foreshadowing or preceding the changes in gene expression that are depicted here by the color of the cytosol of those cells. And what you can appreciate in this example is that we are only able to discern this intermediate cell population by looking at the data jointly. Another promise that this data holds is providing insights into the relationships between the different modalities. However, such novel data also requires novel integration methods, and what I mean by that I will explain to you in a bit more detail. So on the one hand, we're facing the horizontal data integration task, and you might know this problem under the terminology batch integration. Here, we have measured the same set of features, for example, mRNA expression levels, but we have measured it in a disjoint set of cells. So we have multiple samples that were obtained, for example, on different days or in different centers, different labs. And the integration task revolves around removing the technical variation between those samples and retaining the biological signal. When we're considering increasingly complex study designs, it might be of interest to be able to include prior knowledge on the relatedness between samples in our integration framework. So for example, for treatment control experiments where the treatment could be, for example, the disease status or perturbation, it would be good if we could use this information to help us disentangling technical from biological variation. Considering that we're dealing with multimodal data, we also face the vertical data integration task. So we have measured all these different modalities that don't share features. They have very different dimensions, they follow different statistical distributions, they have differences in information content that can be both of technical or biological nature, and they have differences in the data quality. However, we've measured them in the same set of cells, so we know the cell-to-cell pairing between those measurements that we obtained. And here the integration task really revolves around how can we make most of this data and how can we leverage information across these modalities. And I'm trying to solve both of these problems at the same time. And to this end, I developed LIAM, which is an acronym for leveraging information across modalities. What LIAM does is that it learns a joint low-dimensional representation of two single-cell modalities, and while it does so, it can account for nuisance variables such as, for example, sequencing depth and more complex batch effects. LIAM is based on the variational autoencoder framework, and such frameworks have been very successfully employed for unimodal data integration in the past, and we draw from the insights gained from these unimodal cases. LIAM currently supports pairwise combinations of gene expression, chromatin accessibility, and cell surface protein markers. And I won't have the time, unfortunately, to go into the details on all the modeling decisions that I took, but I want to highlight a couple of them that I consider of particular importance. So for such a framework to work, you will need to choose data type specific loss functions. And I want to highlight in particular my use of the negative multinomial loss for modeling the chromatin accessibility data that was introduced earlier this year by my former colleague Wolfgang Kopp. And secondly, I want to give you a bit more insight into the strategies that I employ for removing batch effects. So on the one hand, I employ the conditional variation autoencoder framework. And what this means in practice is that we will feed the one hot encoded batch information to the layers of our encoder and our decoder. And you can think of this as some way for the model to bypass the need of learning any type of batch related information. So this way, you encourage the model to learn batch independent data representations. On top of that, we make use of another strategy, a batch adversarial training strategy that again is inspired by my former colleague Wolfgang's work. So here we couple the conditional decoder and we introduce a novel neural network, batch classifier to be precise. And during the training, this batch classifier will try to predict the correct batch from the low dimensional data representation. And we set up the objective of our overall model in such a way that the objective of this batch classifier and the total objective will compete. And on top of that, we make use of a kind of negative feedback loop through a gradient reversal layer. So when the batch classifier gets better at predicting the batch, we will feed this as a negative feedback during the training to our encoder. And this way, we further encourage mixing of batches. Alright, so let's look at Liam's performance in practice. As a first use case, I brought along a competition benchmark dataset from a competition last year hosted within the NeurIPS conference. So the name of the challenge was Multimodal Single-Cell Data Integration Competition, and the challenge organizers have really generated and curated a large-scale dataset of the human bone marrow. So they profiled bone marrow mononuclear cells, and they set up their study design in such a way that it introduced nested batch effects. So you can see here they've measured samples from different donors across different sites, and they tried to measure one of the donor in all of the sites, but they had some technical dropout here. So you can appreciate that they measure like the dots is multiome data, so the same case that I explained to you before, and the triangles is site sec. I'll focus on multiome here. In total, all of these individual datasets comprise 70,000 cells. And one of the tasks in the challenge was exactly the problem that I'm trying to solve. And Liam ranked fourth for this multiome data in the online training category. And the results that I show on the following slide are a follow-up analysis of mine after the challenge. So what you're seeing here is two-dimensional UMAP representations of the low-dimensional data representation obtained by the different models. And each dot in these UMAPs is a cell, and we can color them by the meta information that we have about them. So in this case, I plotted the sample ID. So the cells are colored by their sample ID. And here I juxtapose the embedding obtained by Liam with a competing model MultiVI, which is a model for mosaic integration, of which this paired multimodal integration is a subcase of, and the challenge winner. And what you can potentially already appreciate from looking at these UMAPs is that we see a stratification in the winning submission by sample. And this is a bit surprising, or what came as a surprise was that this model performed actually best on one of the batch removal metrics in the challenge. And we paired up with the challenge organizers to kind of find out what was going on here. And the reason for that is that this metric is confounded by the nested batch effect structure of the data. The reason for that is that the metric is based on the average silhouette width, and it only ever considers the closest neighboring cluster. So if you have samples from the same site mixing very well, you might be able to obtain a very high score here, even though the mixing of samples between sites is not very well. And this is becomes even more striking when we plot the site on top of these UMAPs. Luckily, there is quite an easy solution to this problem, which is to simply choose a different hierarchical level for scoring the batch effect removal, or use orthogonal metrics that are not susceptible to this confounding, and both of them are shown here. Once we just measured the same metric on the meta-information side, and the other one is an orthogonal metric on the sample. And both of these metrics really clearly demonstrate that both Liam and multi-VI outperform the challenge winner on the nested batch effect removal. A second aspect that I want to highlight is that, so here you see like the color now represents the cell type that were provided by the challenge organizers. And there were 4 metrics that were measuring bioconservation in the challenge, and 2 of them are dependent on these cell type labels. And on both of them, Liam outperforms multi-VI and the challenge winner. So in summary, Liam showed the best bioconservation while successfully removing nested batch effects here between our 3 models on this benchmark dataset. And with this, I'd like to move to the second use case. And this is really where the framework that we set up comes to its full potential. It's when we can utilize replicate information. So the data set here is what I kind of like teased upon a bit earlier. So we have a treatment control experiment with replicates. And this data set is publicly available. It comes from a technology called DogmaSec, where the authors simultaneously measured gene expression, chromatin accessibility, and cell surface protein measurements. And for one of the replicates, they also measured the mitochondrial DNA. I only used gene expression and chromatin accessibility for the training and the cell surface proteins as a holdout modality. The experiment that was performed is the in vitro stimulation of T cells, so this is our treatment. And this particular setup really allows Liam to disentangle biological from technical variation. To showcase this, I've trained 2 variants of Liam. So I actually trained my model twice. And once I used the replicate as the batch variable, and in the other case I used the sample as the batch variable. So usually when you don't have replicates, this would be your setup, like the sample as the batch variable. So this makes it very difficult to disentangle technical from biological variation, because you cannot really attribute the changes that you see to one of these 2 sources necessarily. So what you can appreciate here is that when we look at the replicate, both of these models really mix the replicates very well. However, when we look at the sample, we see that these representations really differ a lot, and we see a very strong stratification by treatment in the representation obtained with the model using the replicate as the batch variable. And I'll show 2 different cell surface protein markers, the expression of them on top of that, to exemplify a bit what is going on. So upon the treatment, a novel cell type emerges, or like a very different function of some of the cells emerges, so activated T cells emerge, and they are marked by CD96 expression. And both of these models actually pick them up, and you can see that they're pretty confined in their expression in the representations. However, there's also secondary effects of the stimulation on other cell types. I think it covers almost all the cell populations present in this sample. And this, you can see, is only reflected, so CD32, for example, such a marker. And you see this very strong stratification into subpopulations, or different populations. The cells are picked up as different population in the model where we use the replicate as the batch variable, whereas we see a total mixing of them when using the sample as the batch variable. And this really demonstrate how, by using these distinct batch variables, Liam can also retain very subtle biological differences between samples. And it's something that one should really take into consideration when trying to integrate the data, because all the downstream tasks, or most of them, they will rely on this initial low-dimensional data representation. To put this into a bit of a bigger picture, I'm currently working on a preprint in which I also discuss which of the modeling decisions are most influential. On top of that, I've been working on avenues for evaluating vertical integration and how to further enforce batch effect removal by tuning the adversarial training strategy. For future work, I'm thinking about including more modalities and extending the framework for mosaic integration. And with this, I'd like to summarize that Liam can simultaneously solve the vertical and horizontal data integration task. It showed very competitive performance in a recent benchmark while successfully removing the nested batch effects. And its flexible framework can exploit replicates to highlight different aspects captured in datasets. And this is of particular importance in light of the dependence on downstream tasks on the embedding. And with this, I'd like to thank all of you for your attention, and I want to thank my group, in first place my supervisor Uwe Ola, as well as my former colleague Wolfgang, Remo, ZPD and Anna, for supporting me conduct this work, and my funding buddies. And with this, I'm happy to take your questions. And for everybody at home, it will probably be typing in the chat. I'll briefly show you my references in case you want to look up anything.